And third, atypical bacteria, it is a mycoplasma, and actually it's better to identify it as surface parasite or extracellular parasite, in the contrast with previous two, which obligate intracellular parasites. It is a appearance of mycoplasma in electron microscope, and actually bacteria nowadays again is reclassified into different genus. They all belong to one species, Mycoplasmataceae, and previously they were described as a single Mycoplasma genus, but nowadays it is Mycoplasma and Ureaplasma. So by nature, again, bacteria is gram-negative, but routine light microscope will not allow you to identify the bacteria because of very small size. Bacteria is motile, but in the contrast with other bacteria type of material, it is unique. It is gliding by the surfaces instead of presence of regular pili and flagella. Uh, Bacteria is usually described as facultative anaerobic, but actually we have some species which have obligate anaerobic metabolism, and nowadays it is the smallest microorganism which is capable of self-replication, independent and complete. So main difference why mycoplasma is separated from ureaplasma is metabolic activity. Uh, so mycoplasma can metabolize glucose or arginine and cannot metabolize urea. And ureaplasma, as it obvious from the name, can utilize urea, and it is the only uh, significant reason to classify microorganisms into two different genuses. So a lot of species are described, and actually about 16 of Mycoplasma and Ureaplasma species, actually Mycoplasma species, are identified in different systems of human body, and they colonize in our surfaces without any damage. Mycoplasma have unique structure. It is the only microorganism which is naturally lacks cell wall, and it is due to unique structure of DNA. It lose genes which are coding cell wall structure, and by that reason, bacteria needs a host cell because actually instead of cell wall, bacteria have three-layered and very thick membrane, and that membrane contains sterol. But sterol is very difficult compound which requires multiple genes for coding, and mycoplasma have no enough space in DNA to code that element, and actually synthetic metabolic properties of bacteria are not sufficient to synthesize that compound. So bacteria can only take that sterol from environment, and usually that environment is some human body. And that's why bacteria needs to stick to the surfaces of the cell, because cell wall of surface of the cell in eukaryotes is rich with sterols, different types of sterol, including cholesterol. And naturally, bacteria have spherical shape, but actually, according to different conditions of environmental surrounding, it can change the shape. If it will be compressed, it can take some triangle, oval, or any other form. We have, similar to chlamydia, two main types of diseases. First one is respiratory disease, and actually it is known as a walking pneumonia because people usually have so mild symptoms, they will not be hospitalized, or will not have a bed regimen, they will have common way of life. Causative agent is Mycoplasma pneumonia, and similar again to chlamydia, disease is identified only in human, and transmission is always human to human contact. The main affected population is kids up to 5 years of old, because they always have prolonged contact, close contact, and kids collective in kindergartens. And disease can be transmitted by air droplets or by different types of close contact, by some sharing toys and playing. Uh, so bacteria have affinity to respiratory epithelium, and it develops specific organelle which helps to attach to cellular epithelium of respiratory tract. Bacteria is very good fixed here to epithelial cells, and it cannot be removed by activity of cellular epithelium, by coughing and by other natural mechanisms of protection. Besides that, recently it was identified that bacteria can produce unique toxin, unique viral factor, which is identified as community-acquired respiratory distress syndrome. It is a toxin which, during prolonged secretion, leading to inflammation of respiratory epithelium and dysfunction of our airways. And in classic way, mycoplasma living on the surface of the cell, but actually it was identified that bacteria can invade deeper in the tissues. By endocytosis, it can perform intracellular replication. So again, some authors, some scientists describe that bacteria is intracellular as well. If you talk about main course, mainly disease is classified into two clinical syndromes. It is tracheobronchitis, which is usually called as a chest cold. It manifests with common symptoms of bronchitis, such as sore throat, cough, some slight fever, and common malaise. Rarely, disease can progress to pneumonia, and again, symptoms are not specific, regular symptoms of pneumonia, but similar to chlamydia, duration is very latent, prolonged. Cough is rare, sputum is rare, and usually not mucoid, and disease is difficult to identify because acute symptoms are not present. Second type of disease, the same is non-gonococcal urethritis. It can be caused by multiple species of bacteria. Usually it is ureaplasma, Mycoplasma genitalium, and Mycoplasma hominis. So disease is transmitted only by human-to-human contact, and usually it is sexual contact. Disease can occur as acute, chronic, or chronic with recurrent duration. It is initially urethritis with very poor discharge, which is mucopurulent, maybe watery, but it's not very resembles real bacterial infection. And again, similar to chlamydia, disease is not dangerous by itself, but complications are extremely dangerous because bacteria can, by that glycerine motility, can move by the way of urinary channel, and it can affect internal organs of genitourinary system. In women, it will lead to cervicitis and pelvic inflammatory disease, and in men, it can be associated with prostate cancers, and again, in women with ovarian cancer. The most significant part of that infection, that in women during chronic and prolonged duration, bacteria can lead to different problems with pregnancy, and if pregnant woman will be infected with that bacteria, mycoplasma can infect different tissues of placenta and fetus, and can lead to spontaneous abortions, premature or stillbirth, and can seriously impact outcome of that pregnancy. So several problems with identification of bacteria. Again, it's so small. It is again the smallest microorganism. It cannot be seen in light microscope, and usually specific devices are required to find that bacteria. And cultivation is extremely poor, and in liquid medium, if it is some specific medium, it cannot produce visible turbidity, so it's not possible to identify and to see is it growing or not. We cannot evaluate properties of that growing culture. Cultivation, actually, mycoplasma is that exception. Some species of mycoplasma, especially ureaplasma, can be cultivated in culture medium, but again, it is so specific culture medium that routine labs don't have it, and again, culture method is not possible for identification. And usually, we will use PCR as the main technique, or again, that rapid test can be applied. And if you remember topic of antibiotics, we have a massive class and the biggest class of antibiotics, such as beta-lactam antibiotics, and they all are targeted against cell wall of bacteria. And of course, if mycoplasma naturally lacks cell wall, that group of antibiotics will not work at all because they have no target for efficacy. And it is important to know because diseases caused by mycoplasma should not be treated with any type of beta-lactam antibiotic. And we have macrolides and tetracyclines as the main agents to treat mycoplasmal diseases. So the same diagnostic measures, we have a ELISA test to identify antibodies or bacteria itself in clinical specimens. And we have rapid tests, which are based on immunochromatography technique. Usually urethral and cervical swabs are used for those tests. Principle is the same. I will not explain it for the some fifth time. And possible problem is resistance to antibiotics, and of course, the main question is how to evaluate efficacy of antibiotic if cultivation of bacteria is not possible. So we have some commercial test system. It is one of them. It is called Mycoplasma IST3. It is a specific plastic strip with multiple wells. As you can see, they are assigned a number, and different solutions are added here. You need to inoculate clinical sample. It is actually a specific test system which will indicate you biochemical activity of bacteria, can help in identification of microorganism. And it contains in the right part of both zones, contains zones with antibiotics. LVX is levofloxacin, MXF is moxifloxacin, tetracycline, erythromycin, and clindamycin in the case of right part. And actually, that system allows you to identify bacteria and to evaluate efficacy of antibiotics very high and very fast. So that system can be used for evaluation of current clinical case. But routinely, again, PCR is used, and the only result you can have is identification to species. So that's all for current lecture. See you in further lectures. Bye bye.